plots. Now, this first block was not just headlined of what is age and aging and what will it be, and on a social and scientific and artistic level, but now we will leave that level and take a look at hard facts, or naked facts really, of organic life, and you will soon see that naked can be understood literally. Now there are two speakers, international speakers, from an excellence cluster of age research at the University of Cologne, of SECAT, the research center there, Professor Hoppe, and he's part of that cluster, and he has the Hoppe Lab at the Institute of Genetics, and Jane Resnick, Dr. Jane Resnick, who opened in 2013 the Interdisciplinary Research Center in Australia. And now this center, on a mathematically scientific basis and the medical basis, and their university and non-university findings, and there's some institutes that are part of that, and that's why it is called the Excellence Cluster. Not because of a generic or genetic reason, but because of the content of this. We will have a man take the floor first, and he will now talk about neurological neurobiology, and Professor Hoppe knows what he talks about. I expect so, because he was once asked to work in Hamburg, and he was awarded lots of national and international awards. His topic is why we age and for how long. Now, what I understood is that his protagonists are proteins. However, and what their names are, where they live, and how you reach them if you need them, he will explain to you. The floor is yours, Thorsten Hoppe. Well, thank you very much. Thank you to Ms. Fang and Mr. Franz for inviting me to this symposium. And also thank you for the moderation. As Mr. Grabowski has just said, we will now talk about the scientific part of today. Mr. Kruse has actually given a really great introduction, but he's right, it's not just science dealing with old age, but it's a multi-layered topic, and that's why I'm very happy to learn new things here also for my research. Now here you can see our research institute. It's the SECAT Excellence Cluster. At the end of my presentation I will talk more about that, what Excellence Cluster means and who's part of that. However, this research building was created due to the German Research Association in 2007, and also the University of Cologne wanted to join this topic, and that's why it was built. We get the necessary funds to make research on the biology of aging, and today I will give you a general insight into what that is and how you can do research on aging, and how can you describe it genetically, how can you determine it, and what can we do with our findings, our medical findings, and how can we put them into medical therapies. Now what you see here is a painting of Lucas Cranach from 1546. He painted this painting right before he died. He was older than 70, which at the time was much longer than life expectancy was in that century. And it actually summarizes what we just heard. It's the desire of today's generation to be young, even though you are old, to have a rejuvenating measure. I don't know if you can see it, but the person in the left These are old people. You can see that if you zoom on in, if you will. And when they walk through this fountain, they are rejuvenated. It's the fountain of youth. And on the right-hand side, the people are young. And that's what modern medicine or modern society tries to copy, tries to establish by a certain type of clothing, social life, and medicine. And also advertising tries to suggest that we don't have to be old in old age. We can stay young even if we're old. That's not always the case. As you've just heard, there are also some disadvantages of old age. I will talk about that later on. But something what's good to know is that the average lifespan increases continuously. This is a result of medical research because we have much better healthcare than 100 years ago. Diseases can be treated and cured in a better way. And also the economic situation, the nourishment, the work situation is improving. And in the Western world, you can see this tendency mainly, but also globally. You can see that on this slide, and this leads to the fact that we are facing social challenges, of course. In 2050, a third of the world population will be 60 years and older, and that is a huge demographic change, posing huge challenges. And you can also see in the international comparison that people with their countries with people with a higher lifespan due to several aspects, which we can only assume. Maybe it's the lifestyle, attitude towards life, the medical care, and so on. They all play a role, these aspects. Now, this is the background. Now, this brings me to the hard facts, or biological facts, really. Aging is something that we all aim at. We all want to have a long and fulfilled life, but we know that this is not always the case. Mr. Kruse has hinted to it. There are several age-associated diseases, and these are not positive aspects of life. So there's a high risk of dementia, kidney disease, chronic kidney disease, cancer, of course, is a risk. It's also part of old age. The older you get, the higher the risk of getting cancer is. Then there's stroke, diabetes, osteoporosis. These are just a few age-associated diseases that you all know, of course. And very often, you won't just catch one disease at a certain age. 50 plus, here it says you may have an incidence of 2 and more conditions. It's the multimorbidity, that's what it's called. So it's that you have more chronic diseases, a higher incidence of these conditions. And medically speaking, we try to find the root of these conditions. And of course, in medicine, these diseases are specifically dealt with. Now you've got a classical approach with specific treatments depending on the disease in the clinic. So for example, if you have cancer, cardiovascular disease, then there are different treatments necessary. However, when we talk about aging as a complex system, which enables old age diseases or age-associated diseases, and they can come up with a new approach. This is something that we do research on at Secund, that we try to avoid age-associated diseases. If we were to find out what leads to aging and aging processes enabling such diseases. So what happened if we were to change our situation in old age? Could we then also reduce the number of age-associated diseases systematically and prevent them? Now, if in old age you have a lower biological age than your chronological age, then you probably are less apt to have an age-associated disease. So that means we need an understanding of the biology of aging. So why does our hair get gray? Why do we have certain physical conditions? Why are we more fragile in old age, the older we get? What determines our age? And here the first studies have been carried out in the 1990s with the model organism that I'm showing to you right now. This model organism is probably not known to most of you. This is a 1 millimeter worm living on the grounds or in the grounds. It's called C. elegans. In the 1970s, it was found by C.T. Brunner, and it was used as a model organism because you can manipulate its genetics, and it was studied by these biologists. And this worm looks quite boring, maybe. Not very exciting, very simple. It's just 1 millimeter long. If you are in your garden going through the soil, you will probably see millions of such worms. So you've met him before, but I've a quote from Friedrich Nietzsche, who of course did not think of C. elegans, but he summarized actually the whole prospect. There are many similarities between this worm and humans. They are genes that are highly preserved. That means genes that are not just existent in worms, but also in humans, and they have the same effects. And Cynthia Kenyon used these findings. She's a pioneer of age research, and for the first time, with the help of this worm, she could manipulate age. Up until this point in time, it wasn't completely unclear what our aging process is based upon. And we didn't have any graspable ideas of age. Of course, nutrition plays a role, genetics, hereditary aspects. But Cynthia Kenyon showed that it's quite easy. She just used mutations on just one single gene. And in C. elegans, that leads to the fact that the worms live 3 times longer than usually. So what she showed here, that's what we called lifespan curve. Now here, each dot is one of these worms, and she had a look at how long does it take until the worms die. So it's just a general worm. It dies after 20 days, so the lifespan is 20 days, which is not very long. It's just about reproducing. A worm can have 300 ascendants, and then their task is done, if you will. And if you compare it to the maximum age, 20 or 25 days, and you can very well imagine that the worms that were examined on in this stage of life, they looked quite old, their muscles were degenerated. Many of what we know from humans happens also in the old worms. Now, this stem here, the mutation of a certain gene, which is called DAF2, which is not very important, it's just a gene. And here the worms remained or appeared much younger. They moved better, they had less degenerative processes, and the worms died only after 60 days, which was quite revolutionary at the time. Now, if we were to adapt that to humans, that would be dramatic. By just a gene mutation, we could have a 3 times longer lifespan. And what I described earlier, it's not just a 3 times longer lifespan, but actually we could have a much longer time in life where we are young. And here, I don't know if it works with the video, if we have the technical means. Well, actually, this would be a worm which lives for just 20 years, so it's almost the end of the after 20 days, almost right before the end of its life. So it doesn't really move much, doesn't eat much. This worm is just as old on the right, but it has the mutation in the genes, which leads to the fact that these worms live 3 times longer and they are much more active, can move, eats. And that would actually be the situation where they have much less age-associated diseases. So one gene mutation makes a huge difference.
Jetzt irgendwie zum nächsten Bild kommen.
I'd like to go over to the next slide. Right. Now, what the San Francisco researcher found out with C. elegans was very revolutionary, because this was not just true for the worms, but also it is a hugely preserved phenomenon. Preserved means this is not just true for worms, but for other organisms as well. And many organisms are being used in our Sickert lab. We examine them. Now here you can see the worms. And here's the Drosophila. It's a fruit fly. You know that when you have mature fruit in your kitchen, then you can see all these fruit flies flying around, quite nerve-wracking, if you will, quite annoying. But the same gene, which leads to the fact that worms can live 3 times longer and fruit flies live longer as well. And the same gene, if it's mutated, then mice will live longer as well. So there are genetic mutation processes that can steer the aging process in different organisms. Of course, we cannot do that with humans, because gene manipulation in humans is ethically not allowed. However, what we did, we had twins, where both twins were older than 100 years old, and they examined them. We examined them, and we used the genome, and we found out that with the same gene that we used in these model organisms that led to a longer life, also led to differences, to changes. So these are preserved genes in different organs, and even in humans, in different organisms, even in humans that would lead to a longer lifespan. And these twins who became older than 100 years, and in the past 20 years in life, they did not experience severe differences. Only just right before they died, they had a sudden appearance of disease or symptoms. But up until then, they had quite a healthy life, similar lively as the worm that I just showed to you. Now the question is, what can we learn from these genetic studies? We cannot have human genetic manipulations. That is forbidden. However, can we learn something from these processes? Can we use that differently to have an effect on aging? And one mechanism I'd like to show to you is calorie restriction. I don't know if there will be food in the break, but maybe that is something we can deal with, whether you should have the biscuits or finger food carrots. What should we choose, vegetables or sweets? So calorie restriction has something to do with nourishment and eating, if you will. And we did an experiment with different small organisms, model organisms. So what happens once you give these model organisms less calories than they would usually take in if they were given the opportunity? They still were sufficiently nourished so they wouldn't starve or lose weight. That's what we call calorie restriction. So for humans, that means not 2,000 calories per day, but 1,200, which would already lead to the fact that we would still be hungry, but we wouldn't starve. And with C. elegans, or the fruit fly and the mice, that would lead to the fact that they would have a longer lifespan, an increased lifespan of 35%, just because they reduced their calorie intake.
So this would be an option for all of us to promote our health, our cardiovascular system. The values that we got are so great, you can never reach those values with physical exercise only. And if you have to have surgery, your regeneration after surgery has proven to be much better if you've underwent a calorie restriction before. It is possible to do that with several different organisms. And as you can see here on the list, there are also bigger animals, mammals, there are dogs. There is a study that currently is looking at dogs, a study from the US. And here, 2 different studies were conducted with chimpanzees. These 2 chimpanzees are of the same age, fully nourished and with calorie restriction. I hope you can see that on the pictures. The difference, it becomes very clear. The chimpanzee which did not take so many calories in looks much better. When we look at humans, calorie restriction leads to a reduced body mass, of course. Fatty acids are less, risk factors are decreased, less occurrence of diabetes, arteriosclerosis, and you have more energy in general. So, calorie restrictions for human beings, that would lead to leading a more healthy lifestyle. Obesity is a risk. What we need is the optimum balance. Of course, an undernourishment is dangerous. You have to balance it carefully, and it might vary from person to person. It depends on how much energy your body consumes. A healthy diet is important, but also you need physical exercise, and I believe all of that is becoming clear to each and every one of us these days. Now, the idea, of course, behind this is to find pharmacological substances which convey to our body that there was a restriction in calories without there actually being a restriction in calories. So some of those substances can prevent age-related diseases, bowel cancer, neurodegenerative diseases, etc., etc. Another process with regard to aging, which is vital, is the difference between germ cells and other cells. Germ cells produce specific types of cells needed for reproduction. So regardless of age, these germ cells basically lead to the existence of new organisms in reproductive health, and that is a rejuvenation of cells which then undergo aging. So these germ cells are passed on from generation to generation while the somatic body cells age. And then people found out how to take away germ cells. If you take away the somatic cells, you can prevent aging. The production of germ cells, which helps in reproductive processes, they will need energy. So somatic cells becoming older, aging, lead to a situation where your body doesn't have enough energy anymore. Correlation with human beings. These are studies that have been conducted with eunuchs. And they have shown that male persons that are eunuchs live longer. So apparently there is a correlation. In these male study objects, the testicles had been removed and they lived longer. Then we looked at hormones, growth factors, etc. So systemic aging, that was the next field they looked at. If you look at tissue, the aging of tissue, endocrine factors, here you see a study from conducted with an Andean family in Ecuador. They are very small in growth because there's a receptor missing that is a receptor for growth hormones. This family, they have a higher risk of liver cirrhosis and a higher risk of other factors, and the growth factors also have an effect on life expectancy. So growth hormones are decreased in number in old age. So studies are ongoing to find out whether you can influence the aging process via these growth hormones or growth factors. So these are the three interventions, potential or possible interventions for healthy aging. Calorie restriction out of the three presented is the easiest one. Each and every one of you can try it to see whether it has an effect on you, what it feels like, and to test whether it and how it improves your health. That is one option to be very pragmatic. Which conclusions can we draw then? Somatic cells age, however, germ cells are stable. Our genetic pool was not selected for extreme longevity. The accumulation of damages in old age has a negative impact on the function of cells and tissue. Genetic pathways regulate longevity, and as I've shown, these genetic pathways exist in many organisms. Nutrition and lifestyle have an influence on aging and age-related diseases. There are interventions which may influence the rejuvenation and maintenance of cells according to cell type, and it's necessary to have a paradigm shift in medicine to not treat the disease anymore or the disorder, but to maintain health. That should be the new objective of any therapy. So, final question, how old can we get then? Which age are we able to reach? It's difficult to answer, however. We can see that life expectancy is increasing continuously. In the Western world, it's about 80 years right now. The oldest person that ever lived is a French woman named Jeanne Calment. She died when she was 122 years old. She smoked, she drank a glass of red wine every night, and at the age of 110, she starred in a music video of a German band even. She featured in the video. She was born in 1875. Back then, the average life expectancy was 43 years. Researchers in Singapore, Russia, and the US have recently calculated, just a year ago, by computing our life expectancy to theoretically be up to 150 years of age, if everything works smoothly. Let's briefly look at our research center. It was established in 2007. And we try to look at the biology of aging to understand it, not to use it to become 150 years old, but to use it to do research in the prevention of diseases, for example. We have different research clusters. It's a research cluster because numerous institutions are involved. This is CCAT in Cologne. Right next to it, it's the Max Planck Institute for the Biology of Aging. This went hand in hand with funding from the federal state and also the federal level. We have molecular medicine, we have genomics, the animal facility, metabolism research. All of those work hand in hand. So over the past years, we have made a small contribution to the research I've presented to you today. Thank you.
Und ich danke Ihnen, Professor.
Thank you very much, Professor Hoppe, for this presentation, which was, from your point of view, maybe even a bit superficial, but for some of us, it was amazing to learn the current state of life sciences aging research, in particular this part of it. There are moments in time where you have a personal affection for a certain topic. When you set calorie restrictions, well, when I look at you in the best of all shapes and compare it to me, well, you clearly embody what you preach, so I like that. It's wonderful. And